Hello, hello. Good morning. Good morning, everyone. This is Oscar, and I have the pleasure today to be with Professor Ali Mobasheri, sorry about that, a globally recognized leader in osteoarthritis research and cartilage biology. He is a professor, a scientist, a leader, and a critic. Today, we're actually going to be talking about a lot about different topics in regenerative medicine, stem cells to gene therapy, autologous and allogenic. And we're actually going to be talking about a little bit about regulations as well. Ali, how are you today? How are you doing today?
Good morning, sir. Nice to see you again. I wish we could have done this face to face in the same.
Yes, yes. We will one day. We will do one day for sure.
But thanks for having me. I've been following you and listening to a few of these you've done, and I'm looking forward to seeing how you can expand and um, reach out much further into our scientific clinical communities with really topical discussions in regenerative medicine.
Yes, thank you. I obviously saw, I've been following you as well, and I know that you liked a couple of my posts, and I reached out and I'm like, I would love to talk more, openly and have these discussions with scientists and leaders like yourself, other physicians. And I also would love to host also, other doctors and, obviously have, head to head doctor talk as well and scientists as well. It's very much needed. It's something that I see that the industry needs in regards to the nuance. There's so much in the nuance that gets lost, right? And a lot of even people myself, that's why I've been reaching out and looking for answers and really understand regenerative medicine as a whole, right? So I really appreciate you doing this with me.
Not at all. I think that nuance is very important, and we're going to touch on various nuances when we talk about some of the topics today. For example, stem cells, regenerative medicine, gene therapy, cell therapy. Um, exactly what are we talking about? What type of cells? What kind of platform? Which genes? And we will try to minimize the use of jargon and abbreviations that some of your viewers may not fully understand. So stop me and tell me to define things. But I'd like to begin with a pitch, and I want to be provocative and say, look, 20 years ago, we were made a lot of promises about stem cells. Oh, yeah, stem cells are going to go in the joint and regenerate the joint, and we're going to have a fully functional knee again. None of that was delivered. None of that actually happened. And we now think of stem cells in a very different way. We think of them as paracrine, think of them as immunomodulatory therapies, transient. They don't last forever. They may not even last beyond a certain number of days. We will talk about that in more detail. And we need to move away from this stem cells are going to cure. Cure is a very complex word, and we mustn't use it. I'd like to propose different terminologies for you. I want us to move away from simple stem cells to an era of molecular precision orthopedics, where we have a number of orthopedic and orthobiologic platforms that we can apply to different people according to the kind of phenotypes that they may have. And that's, I think, the exciting thing about the future. And as we move forward towards the future, we're going to make some mistakes. And it's important to make mistakes.
That's how we learn.
I hope some of my students and some of my team members are listening. That's how you learn and grow and get better. You have to make mistakes, and to understand how to do things differently, we have to learn from those mistakes. And in medicine, in regenerative medicine and orthobiologics, we have made and we will make a lot more mistakes. So let's move forward with the recognition that mistakes and learning from them is really important.
Yes, I agree with that. Even in my entrepreneur career, I made so many mistakes, right? And that's how I learned. I'm like, okay, I'm not going to do that again. And then I try to find a different way and I fix it, or sometimes I make another mistake, but I learn, right? Until I get it, until I get it done, right? So that concept can be applied pretty much in everything in life, right?
Yeah. If you did ice skating and if you learned how to ride a bike, you're going to fall off the bike and you're going to fall flat on your butt quite a few times on the ice until you get it right, until you learn and master how to control your balance on a bike, on skates, on rollerblades, whatever you're learning. And that is an important part of learning and moving forward.
Yes, I agree. Okay. So, a lot of things that I see, on LinkedIn is we were talking about the nuance, right? And you mentioned also about that you want to, stay away from like the word, stem cell, be more paracrine and more precise, right? Can you kind of elaborate a little bit about that? And what do you mean by that?
So, stem cell and the promise of stem cells didn't really deliver, right? We are waiting, and there are lots of platforms, and some of them have actually, reached phase 3 clinical trials. So in osteoarthritis, there's a couple of recently completed phase 3 clinical trials of mesenchymal stem cells, MSCs, and they didn't prove to have any impact on any meaningful clinical impact on pain and function. But there's other cell therapy platforms that are ongoing and being investigated in phase 2 and phase 3 studies. And we need to wait a couple more years for the results of those. But I want to again take you back to 20 years ago, 30 years ago, in the late 90s, um, when I was finishing my doctorate. You open up Nature or Science, the covers basically tell you the whole story. The promise of stem cells, the promise of regenerative therapy. Now, from a scientific perspective, we have to be very critical of all of our failures, right? But we also need to recognize that for some of the conditions that we're trying to treat, um, in particularly in orthopedics, there are no treatments, there are no approved treatments, and there's a very significant unmet medical needs. So again, I want to use the word nuance is very important, the balance between what is published and what is medical evidence and the demand for treatments and is what patients and consumers want. So let's focus on a more in-depth discussion of stem cells, right? But also bearing in mind that there's two things to balance: scientific and clinical evidence, and also how these things may um, perform in a real life context. So let's begin by highlighting the big differences between a randomized placebo controlled clinical trial, right, of orthobiologic intervention, right, or a drug for osteoarthritis. It doesn't matter. Trying to do that study in a randomized, placebo controlled, double blind, very controlled way in an expensive clinical trial is very different to doing it in the office or the work location of a practitioner who genuinely cares about his or her patients, and there's empathy, and there's a personal relationship, there's warmth. And this is why so many of the real world clinical studies of orthobiologics, stem cells, you name it, um, differ in their outcomes from those very tightly regulated clinical studies. And part of that comes into, of course, they're not blinded. In a real world scenario, patient comes in, there's a discussion about treatments that are available, and there's probably some pre-existing reading on the internet or talking to others, and there's a demand, and then the doctor explains the scenario, and they're told that they're going to receive a treatment, but they've received that in a very caring, in a very empathic way. The outcomes of that are going to be very different from a very tightly controlled clinical trial. We don't often talk about it, but that's another important nuance. And this is why in the area of intra-articular injections with hyaluronic acid or some of the newer formulations of hyaluronic acid, there's all the evidence that says, there's minor impact and very fringe benefits for patients in a very tightly regulated clinical study compared to a real world study. So we need to bear that in mind. And I think the same also applies for some of the orthobiologics, whether they are stem cells derived from the patient themselves. So we're going to talk about PRP, platelet-rich plasma, a little bit, I guess. But let's focus the discussion on stem cells. If I'm having an autologous stem cell treatment, that is stem cells from myself. Okay,
Correct.
I receive those stem cells from my bone marrow or from my adipose tissue, and basically it involves removal of bone marrow, quite an invasive procedure, or removal of some fat and then isolation of the cells from that, and reintroduction. And there's another nuance: is reintroduction immediately after isolation, or reintroduction after some culturing and passaging in a laboratory environment. That's where the regulators have different views on, point of care. So there's the point of care stem cell therapy versus an autologous therapy that goes into a lab and that becomes an advanced therapy. It's not
Yes.
point of care therapy anymore. And there's allogenic, which is a platform. So there's a cell platform, and that cell may have been specifically isolated, characterized, and studied for its molecular portfolio, molecular properties. The secretome has been characterized. We know which growth factors it's producing. We know if it's got vascular endothelial growth factor or um, fibroblast growth factor. We know how much of all the specific growth factors or cytokines there might be in the medium that cell produces. Now, of course, that platform is allogenic, and that is where you can have a therapy delivered to multiple patients from the same individual. So there are a number of ongoing phase 3 clinical trials in osteoarthritis that actually derive from a single donor, um, a cell, and that's an allogenic therapy. Could be genetically um, manipulated as well. And so it could have, for example, a gene introduced into it to overproduce a particular regenerative growth factor. And so these are really important nuances. And so not all stem cells are the same. So some of the stem cells could come from your own body, point of care, injected in the same day without having to be grown in the lab. Other treatments need to be grown in the lab, processed, and then they become an ATMP. Then, of course, there's allogenic therapies that are usually much more industrially produced and controlled platforms. So I think patients need to be educated about what these things are, the nuances and the differences between them. And of course, when there's a debate about whether stem cells work or not, all of the nuances are lost. Which type of stem cell are we talking about? Which trial are we talking about? Was it the trial that was completed 4 or 5 years ago, or the one that was just completed in the summer of 2025? Or are we talking about the trial that's actually not going to report until the late, maybe fourth quarter of 2027? So that is an important area to discuss in a open dialogue with you and with your audience. Those nuances mustn't be lost.
I agree with you 100%. And then one thing too that I've been, learning as I learn and read, all the nuances between, stem cells, right? Either it's autologous, allogenic, is also the processing is very important as well. And it gets lost, right? Now, on the autologous side, you have various, devices, right? That process either, your blood, fat, or bone marrow, right? So, and then you have user variability, user error, and then that's only on the autologous side. Then on the allogenic side, then same thing, right? Like, okay, what are they extracting? What are they, how are they extracting it? How are they preserving it? What are they adding to it? How are they freezing it? So it's just so much, right? And I can, yeah, and I can understand why, it's confusing, right? Even for me, right? Like I even for me, it's confusing, right? So I can understand for patients, for providers that are still getting new into it. So I understand all that.
The other point which is worth discussing is the age of cells, the stem cells, the biological or the chronological age of stem cells. Another very important nuance, rarely discussed and drilled down into really what do these things mean. So
So, um, can you? So we know what biological age is, chronological age is, but can you define that in like simple terms? Like, what do you exactly mean
yeah,
biological versus chronological?
there's a massive difference between biological age and chronological age. So I'm not going to tell you how old I am, but if you took some of my cells out and grew them in culture, in plastic dishes, I would hate for those cells to put into any recipient because I'm older, right? And I think there is a level of senescence, there's a level of aging. Despite my youthful appearance, my stem cells have aged. My cells have several decades. They've clocked up several decades. So, and all cells have got telomeres, and we know that one of the hallmarks of aging is shortening of telomeres, and telomerase activity is one of the key processes that does that. So, what we really want, I think, in terms of treating an age-related degenerative condition, we're not going to talk today about any neurodegenerative conditions. It's not my area of expertise, and we can probably just touch briefly upon the big differences between joints, orthopedics, and Alzheimer's, Parkinson's, etc. But let's focus on orthopedics. So in orthobiologics, in orthopedics, there's a big difference between a stem cell population that is derived from a birth tissue like placenta or Wharton's jelly. That's basically biological age 0, right? Because it's from a birth tissue. The fetus has just been born, and the placenta is a byproduct of that. And you can take cells out and develop, and there are several therapies, even right now in phase 2 clinical trials from placental derived stem cells. And there's a big difference between doing that as a therapeutic versus taking my cells and putting them back into my own joints, or God forbid, into somebody else's joint. Uh, so there's a fundamental difference in terms of the biological age. And we know that with aging, stem cells also age. And you and I both have stem cells too. But with every passing decade of life, there's senescence and there's aging. And older cells are not going to have the same robust regenerative capacity as younger cells. But despite that, there's a lot of resistance and hesitance when it comes to those things. And obviously, some of that comes from the regulatory aspects, from the regulatory perspective. Some of them could be even religious. And again, that's another topic we cannot get into. Let's not get into what different religious denominations might think of birth tissues versus, aged autologous cells. But those are important dimensions that, again, are lost in the translation of these concepts to the general public. So there's a lot of skepticism about stem cells, but it's very important to look at the evidence and determine what was originally injected, what was the biological age of that, right? And what were the real regenerative properties of that? What was the potency? It's another terminology that's very important in regenerative medicine. What is the potency of a particular regenerative therapy or stem cell therapy?
Interesting. You said something there that kind of stuck with me because you said fundamental, right? So knowing that as we age, our cells get older, right? I personally, in my opinion, I don't think you have to be a scientist or a doctor to kind of know that, right? To kind of, or go to school for that. So, but there's been multiple things that I see on LinkedIn and on social media saying that that's not true, right? That as we age, our cells, they so it's just very confusing when I read those from doctors, from researchers that say that as well. So obviously it's just a little confusing and hard to understand what's true, what's not, and then the data, like you mentioned earlier, it's just was it done this summer? Was it done 4 years ago? Who were the ones, doing the study? So there's so many different factors and variations that can change the outcome or the results or anything like that, right? So yeah, okay.
Answer your question, I guess, and to dive deeper into this, we probably need to pick, let's pick Wharton's jelly as a good example, right? Because let's we can agree on the fact that Wharton's jelly is like the gold standard of birth tissues, right? And
I would say.
we know the regulatory agency's perspectives on it, but ESCA published a systematic review. ESCA is one of the European orthopedic societies, and they highlighted that Wharton's jelly mesenchymal stem cells have superior proliferation rates and higher concentrations of a number of anti-inflammatory cytokines and growth factors compared to older, more mature, my age stem cells, adult stem cells. Now, has there been a side-to-side comparison? Has there been a direct side-by-side comparison? No, nobody's done that study. You can take cells, you can grow them in a laboratory environment, and then you can study the secreted proteins, the secretome, and now we have very powerful platforms, mass spectrometry and proteomics, to identify the proteins and quantify the proteins, and then be able to compare the secretome of one cell to another, or one particular platform to another. But that's rarely done, you know. And so very often when you hear these things discussed, there is no comparison to a gold standard. But it turns out that placenta and Wharton's jelly, in terms of what we were talking about earlier, biological and chronological age, they are young tissues. And they're, of course, more likely to have a secretome that's richer in growth factors and anti-inflammatory cytokines. In science and in clinical studies, academics get a little bit protective of their turf. They may have worked with a particular type of cell or platform for a while, and they've built whole careers on that. So they become uber defensive and very protective about any, especially if you come with any criticism pointed towards their platform or their cells. And so that's the challenge, because let's face it, science is ever changing. It's always up to scrutiny. Any scientific finding can subsequently be challenged and disproved. And we've seen a lot of recent debate and discussion about the role of the protein tau as one of the biomarkers of neurodegeneration in Alzheimer's. And
Hmm.
some of the papers that were published, some of the seminal papers that were published, turned out to be fabricated. There's been evidence of some of the papers have been retracted. There's been fabrication of data. So unfortunately, not all of science is always built upon truth and honest principles. So you've just got to take the best evidence that you can, right? And this is why there's so much reliance on the pyramid structure of the randomized, placebo-controlled, double-blind clinical trial as like the gold standard of comparing a new treatment, biological or not, or conventional, versus a gold standard. But there has to be a gold standard. There has to be something, there has to be a standard of care. And unfortunately, in the area of osteoarthritis where I work, the standards are terrible. What are the standards? Corticosteroids, non-steroidal anti-inflammatory drugs. So we're comparing to things that are not actually ideal, and they don't work for extended periods of time, and they may not be actually very safe for very long-term use. So that's the challenge. But ideally, what you want to have is a gold standard to which you compare to. And that's how, you can build on those principles of good science. But I remember 20 plus years ago, we were told stem cells are going to create um, regenerative factories that will rebuild the joints. And
Mm,
I'm still, as a scientist who's been in the business for several decades, I'm still looking for the evidence for that. I'm entirely unconvinced that we have um, cell regenerative factories that regrow new cartilage. Now, there have been some really promising treatments, like you may have heard FGF18, fibroblast growth factor 18. It was turned into a recombinant protein called sprifermin, and the sprifermin was studied in a trial called Forward, and they looked at regeneration. And of course, it regenerates cartilage. There's evidence of cartilage regeneration. And some really good quality papers were published. But it turns out that despite regenerating the cartilage, it doesn't have much of an impact on pain and function. And so the patients still feel pain. And this is
interesting.
why the regulators are saying, well, uh, to qualify for a true disease modifying drug for osteoarthritis, the treatment has to have an impact on pain, function, and um, be able to induce some kind of regenerative, reparative response. Um, so far, we don't have any. We don't have any agents that can do all of these things. It's quite a complicated ask, actually, to a single chemical entity or to a single growth factor, recombinant protein, um, or even antibody to say this one mechanism, this one target can reduce pain, improve joint function, and regenerate. And so this is why there's now so much interest in stem cells and the secretome of stem cells, the extracellular vesicles of stem cells. I guess that's another area we're going to talk about, the distinction between cells and their secreted products, because they're different platforms and they can be applied in different ways. But that is where we are heading. We're heading towards a lot of trials looking at extracellular vesicles and secretome, because that way you don't have to be dependent upon a live, living cell population. Because let's face it, the majority of stem cells that go injected into the joint, they have a limited viability. We're not talking weeks and months. Cells are dead within a few days. There's been a number of studies to show that the cells die very rapidly, and actually some of the inflammatory clearing up of those dead cells could be part of the um, therapeutic response. So imagine,
Is that on the autologous and allogenic
I think
side?
it's probably for both, because the cells, imagine whether it's autologous or allogenic, it doesn't matter. If the cells are dead after 5, 7, 9 days, then the immune system has to clear them up because there's debris from those cells. There's nuclear material. It's a mess, and so that's probably where neutrophils and macrophages and immune cells come in, and they start to clear the big mess. And maybe the therapeutic response of some of these stem cell therapies is associated with that immune response, and maybe that immune response is like a reset mechanism. It's a little bit like, when your computer um, is really ready to hang up and the only way to get it working again is to reboot it. Maybe it's a similar mechanism where the immune system sends in neutrophils and macrophages to assess the situation, mount a resetting response, and then you reduce pain and have reduced pain and inflammation as a consequence. But it doesn't matter which platform you have, allogenic or autologous. Cells die, and nobody likes to talk about it. Everybody makes the assumption. Some of my colleagues working with MSCs, oh, we're going to inject these into the joint. Well, how long do the cells have to live? Oh, it's not an important question. But it is an important question. It's the crucial question. It's the most important question. Because if your cells are dead, uh, then we have to ask the fundamental question. Maybe there's a mechanism of therapeutic intervention that requires nucleated cells, the nuclei and the breakdown products. And then maybe that's different from extracellular vesicles and secretomes. And really, right now, nobody knows the answer. I'd like
Hmm.
I'd like any listener to challenge me, maybe afterwards send me a message to say there's a paper that you haven't read that makes the clear distinction between these, and you don't know your stuff. Please, listeners, if there is that paper, send it to me, communicate with me. You won't find that paper because it doesn't exist. Nobody's done it because we cannot do. These are not easy studies to do. Um, the answer
Why
is
are they not easy studies to do?
because of the number of patients you would need, because of the costs, because of the
Mhm.
complexities of designing such a study and the randomization and blinding and all of that. You got to do it properly. It's very difficult to do. Okay, so you might say to me, okay, that's too difficult to do. Why don't we do it in a real world setting? But what are we gonna? It's like comparing an apple and an orange. You know the differences already. You know they're very different. They're gonna have different outcomes. So what would that kind of comparison achieve if you already know they are very different? We have to build evidence empirically, and I think based on what we've seen so far, there is some promise in the cells, but we have to recognize that there are long-term doubts about viability and long-term doubts about real regenerative capacity. And this is why there's now a lot of interest in extracellular vesicles and secretome and gene therapy, where there's also another nuance, with cell therapy, with this secretome approach or the secretome of stem cells, there are multiple targets that you can hit, but in a relatively time limited way, because that injection isn't going to last. It's not like a vaccination against measles, mumps, or rubella where you have immunity for life. It's a little bit like a vaccination against the flu virus or COVID that keeps changing, and you need to have refresher shots. Let's not get into the debate of vaccination. That's a different matter. But it's about diversity. And so I think that's where the really important lost nuances.
Yeah, and you mentioned a couple of times actually, that, back in 20, 20, 30 years ago, you mentioned that, stem cells were going to, cure or, help with cartilage. And why do you think 20 years ago we still haven't found anything? Is it because the study is difficult, or why do you think we haven't found that yet?
I think it's because we had very, very high expectations that were not realistic in the first place. We were putting them in patients who were quite advanced in their disease progression. A lot of cartilage had been lost, and a lot of the studies are actually done in people who have lost a very significant amount of joint tissue. And it's unreasonable to expect that any kind of therapy is going to go in there and totally regenerate. There are some very exciting new trials being done with stem cells combined with an extracellular matrix. We didn't talk about naked versus clothed stem cells, because the majority of trials that have been done with naked cells that have no protection at all. There's no physical barrier, there's no gel, there's no matrix to protect them. But there are some new studies being done right now where the cells are co-injected with a biological matrix, and that biological matrix provides a level of protection, but also some of its own associated growth factors and anti-inflammatory mediators. So but then that becomes a combination of a cell therapy platform and a um, biological matrix, which, by the way, the regulators will still not fully recognize as an inert medical device, because if it's a matrix that may have growth factors, it's not really an inert platform, is it? So that's where a lot of companies think they've got a very simple device and cell combination, but the regulators will have different views on it. And of course, you and I need to have a separate discussion, maybe as part of a debate with another um, expert from the regulatory agencies or somebody who's had that kind of exposure separately, because it's too complex a topic for today's discussion.
Yes. That's a good point that you brought up. Regulatory, you mentioned that science is ever changing, and it's right now it's going so fast, and regulators can't keep up with it. Um, and you were also mentioned about the demand, right? Because what I see is that here, you have obviously patients know what stem cells are, right? They at least know what's a stem cell, right? Obviously, the nuance, it's more education on that end, but it's confusing for the patients. Um, I forgot what I was going to say. To be very honest with you, I have so many
You
questions,
were
and I
asking about the regulatory perspectives, and you
yeah,
know,
okay,
you live in Florida, and
yes,
in Florida, it's a very unique
it is.
environment because Florida has its own very specific rules and regulations. We know there's a lot of concern out there amongst the US medical practitioners about application of stem cells, the possibility of litigation, and a very small chance of them losing their medical license. Now,
Yes,
Florida is different from other United States territories where you have your own specific rules and regulations. And I've looked up here the environment in um, Florida. Um, in the FDA has a very specific set of guidance about birth tissue products. Um, they are often much more than minimally manipulated, right? They are not point of care devices. I've never heard of a device where you take somebody's placenta and, you know, inject it back into themselves. No, it has to have digestion, processing, and that's where there's a lot of variation in the products that are produced. And it says in my reading and the notes I've made, the 2025, I think it was June or July 2025, there was a Florida stem cell law which allows doctors in the state of Florida to use these therapies specifically for orthopedics, not for any other conditions, under very specific and tight state oversight uh, with full transparency. So they need to say that this is not a cure, right? So the language has to be clear. This is not an FDA approved treatment. This is not a miracle. This is not a cure. And that has to be completely transparently communicated to the patient and in writing. But there's freedom to operate. There's the doctors based in Florida can actually provide the treatment. And so that actually opens things up a little bit for real world studies. So I think some doctors are clearly concerned about losing their licenses, but there's
yes,
that level of nuance and protection in your state. And that potentially provide some opportunities for people to actually do some real world studies and case control studies to advance the field. And I think it's important when we talk about advancing the field that we recognize the differences between targeting a young athletic population versus a geriatric elderly population, because you're going to have different outcomes, whether the injectables are PRP. I mean, what do you think, Oscar? Clearly, PRP, whether it's PRP or stem cells, do we expect somebody in their late teens, early 20s to respond in exactly the same way as somebody might respond in their 70s? I mean, it's to me, it's bizarre, that kind of expectation. Oh, it's the same kind of treatment, so we should be having exactly the same responses.
Yeah, in to answer that question in simple terms from a non-scientist is if I get a cut, let's say a 15-year-old gets a cut versus a 70-year-old that gets a cut, what's going to feel faster?
Sure. And the best studies have been done with bruising. You know, if you're in your 20s, you play college football and you have a bruise, your bruise is going to recover so much faster compared to somebody in the sixth, seventh, eighth decade of life. And we know that with aging, recovery, repair responses are much slower, and they're not exactly the same. So yet, why are we still doing clinical trials with inclusion criteria that may span four decades of life? You know, we might be recruiting people all the way from their late 40s into their 80s. It seems completely balmy and
You
completely
know,
bizarre to me that we
That's
should
a
be
great
doing.
perspective. That's a great perspective. And you know what I the reason what why I thought most studies why the reason most studies gathered that, that older individuals was just because most of them are the ones with actual more pain, right? So that's why. But having a younger generation or younger individuals having these studies would give us a good would kind of answer that question that we're asking, right? Do younger people heal faster than older people as well, right? So
That's my expectation. That's my sort of scientific hypothesis. I would expect a young cohort of college football players to respond better to PRP injections into their knees compared to postmenopausal, andropausal men and women in their 70s. Clearly, totally different populations, different repair capacities, and different lifestyles too. The elderly patients are going to be less likely to be, mobile and exercising. The younger people are going to have a better repair and regenerative capacity because they're younger and they're more active.
yes, I agree. I agree. And external factors are also you mentioned it. External factors are also important as well, right? Depending where they live,
Where
are they
they live, the socioeconomics of their household, their
what
support structures, and
diet?
diet, obesity, cardiovascular comorbidities, diabetes. We know that high BMI and obesity is a major inhibitor of good repair responses. In fact, in many studies, unless there are specifically studies looking at effects of weight loss on joint pain and function, which I'd like to talk to you about that in a moment as well, because there's been a couple of recent studies showing that weight loss has a significant and dramatic impact on reducing pain and improving function in the knee in particular. But in a lot of other trials for other types of interventions, high BMI would be an exclusion criteria. So you're actually excluding those people. Clearly, you're doing that because you're expecting poor outcome from them. And in a real world population where you've got all the skinny and all the average, weight people and all the obese people to treat the same way? No, of course not.
Yeah.
They've got different systemic inflammatory um, responses and set levels.
Yeah, I mean, the way I look at it is it all goes down. We're interconnected with everything, right? If we eat good food, then our cells are gonna receive good nutrients. They're gonna perform, optimally, right? But and then obviously, if we feel good, stress, right? I think that's all plays, everything in our health, right? So, a lot of things is that, and sometimes I see that and I'm like, it's also not all the doctor's fault as well, because sometimes I'm sure patients don't really listen, right? They don't listen to what their doctors say. Hey, you got to eat a healthier lifestyle. You got to exercise. So it all goes down to the patients as well, I think, right?
Also, cultural
Yes.
expectations.
Yes.
In the United States, there is this uh, fascination and almost obsession about healthy aging. And people who are in their 50s, 60s want to be as active and as um, if you like, sporty as they were several decades earlier. And when they can't achieve that, and if they live in a nice area, around Miami and Fort Lauderdale, West Palm Beach, those areas, we know there's a lot of wealth, there's a lot of affluence, and they have the money to pay for not just aesthetics but also experiment with orthobiologics in order to run faster, better, look better in the gym. I don't know whatever motivation they have, but they have the money and they have the ability to pay for it. They've got the residual income and they will spend the money, whether we like it or not. There's that market, and the market dictates what is demanded from the consumer, the patients, um, from their doctors and the private healthcare system. So there are also important distinctions to make between your uh,
Culture.
US federated, non-federated, public versus private healthcare system. I work in Finland, a socialist democracy. It's got a healthcare system. I'm originally from the UK. It has a crumbling national healthcare system, but it still works and is something to be proud of. And it's really important to look after it and make sure it doesn't falter and disappear, because uh, outside of that, then cost becomes a major driver of what you can and you cannot provide for patients. And then, of course, only the rich can afford the very exciting, very promising things, and then you start to look at economic divide between different groups of people.
Yeah. I mean, here in South Florida, people here, there's a lot of people that I remember when I was in college. I had a lot of friends that came from out of state, from different states, and they would always tell me, like, here in Miami, you have to keep up with people. People are always looking good. They would always say that. And then what I've realized here is that people would also prefer to look good rather than fix their knees or walk better, right? They rather pay that than be active, right? Or they look for the shortcuts.
And there are different motivations, and again, it depends on where they're coming from and what their hopes and expectations are. But generally speaking, Florida and the Miami area is very different from many other parts of the world, and there's this constant hunger. Everybody wants to be more active, looking younger, looking better, staying competitive, and so I think you have a very different environment there.
Yes, I agree. Yeah, when we do marketing and we look at what like certain keywords, where to market, Florida is actually number one when it comes to like stem cells, regenerative medicine. I believe California comes in second. Texas is right third, I believe. And then you have some of the Northeast states coming in fourth, fifth, and things like that. But yeah, we're definitely you know in the stem cell game, I would say. And then, that Florida law also helped a lot.
And it'll be interesting to see how that plays out in the coming years.
Yes,
Uh,
it does. So I know you went, so we talked a little bit about live versus dead cells, right? And I want to touch up a little bit on that because that's very important, right? So I asked you autologous and allogenic, and you think that it's both. So if the cells die, right, within, several days, what's helping the regeneration, or what is helping that? Is it the signaling? Why are and obviously we know that all treatments have success and fail rates, right? So what's helping there, right?
So the person who originally coined the term mesenchymal stem cells was Arnold Kaplan. He passed away recently. I think it was about a year ago or more. And of course, towards the later stages of his career, his very distinguished career, very influential scientist working in our field, he changed the terminology. He wasn't calling them mesenchymal stem cells. He redefined them as medicinal signaling cells. And so that redefining exercise was very much a response to the literature and all the evidence about the immunomodulatory capacity of MSCs and really how do they work. So the viability controversy is still ongoing. So many of my colleagues still think just because they've injected MSCs into the joints of their little mice or rats that they're going to live forever and regenerate the joint. Clearly, in human beings, that's not the case. We know that the traditionalists would expect the stem cells to live, engraft, and grow, and regenerate cartilage. But looking for the evidence for that is much harder. So moving forward, we have to be realistic. Live cell or cell derivatives from a live cell. And that's where, again, I've mentioned the secretome and extracellular vesicles. They're easier to make. They're cheaper, especially from the delivery perspective. You know, if you imagine the expectations you would have from just tech and facilities in order to deliver a live cell therapy into people on a large scale, you need to have serious freezers and you need to have the cryopreservation capacity to cryopreserve and then be able to thaw the cells out and deliver them. That requires expensive tech, expensive freezers, expensive equipment, and this is one of the inhibitory factors. So what we're looking for, we're now looking for disruptors. Disruptive innovation is, okay, maybe stem cells, living stem cells, may not be so easy to deliver across different countries, different continents, different contexts. What if we could simplify things? What if the secretome, which may have microRNAs and proteins, growth factors, anti-inflammatory cytokines, maybe that's good enough, and there are ongoing trials to see if these can be efficacious, not just in orthopedics, but also in some of those areas. I don't want to get into neurodegenerative and cardiovascular diseases, but clearly it's much easier to do a trial of that in a knee joint. It's, again, easier to do it in preclinical models, much harder to do in human beings, much more expensive to do in human beings. And then, of course, demonstrating that the ghost cells, the extracellular vesicles, the exosomes work compared to living cells. Again, those are going to be very difficult and expensive trials to conduct. So I think that's where we stand at the moment. What I see happening in the 2026 to 2030 and beyond is the big debates. Bone marrow versus adipose tissue, which one is better? Which one is less inflammatory? In adipose tissue, the FDA's position is very clear. Enzymatic digestion is needed. You need enzymes to break things up, and that's why adipose tissue cannot be a point of care device, right?
Yes.
Compared to some of the things we know exist currently from bone marrow, where you can take bone marrow and inject on the same day. From adipose tissue, you've got to do the processing, you've got to do the digestion, and then cell culture, etc. But I cannot answer your question and tell you definitively whether live is better or dead. Somebody somewhere needs to do a very disruptive trial. They need to be, well prepared for expenses, because it's going to be expensive, and conduct a trial, but it has to be believable in terms of what is compared to, and that's always a challenge. And another bigger problem that we haven't really discussed is the endpoints that the regulators, particularly the FDA, expects. They haven't told us, the scientific and medical community, what those endpoints are. They've told us that, well, we as the regulators want to hear from the community what endpoints we should engage with. And so there's a lot of discussion right now. I'm part of a European project where it actually kicks off next Monday, looking at
Sure. What do you mean by endpoints?
endpoints that you would use in a clinical trial, surrogate endpoints that would tell you whether your intervention's working or not, things that would be really valuable, sensitive things to measure. And in my field, it's a complete mess. We don't have clearly defined surrogate endpoints. We don't have good enough biomarkers. In some other disease areas, in cardiovascular research, there are much better biomarkers. And so we need better biomarkers and we need better endpoints. And with those in hand, we can probably do much better trials. And that's actually some of the things that we are waiting for in orthobiologics. If we had the better biomarkers and the better endpoints, we would be able to do much better trials. I don't know, maybe some of the listeners might be from the med tech sector. Some of the listeners and viewers may be from companies that are developing wearables. I think that's very promising. So these wearable watches or the rings
Yeah.
could tell you agnostically a lot of information where that information is constantly collected in real time and doesn't require boring and complex questionnaires. Now, the medical community thrives on questionnaires. We love questionnaires, and we think questionnaires are really cool because we are researchers and we've developed these very sophisticated questionnaires, and we claim to have them as sensitive as you can get. But ask any patient or patient group, what do your stakeholders think of the questionnaires? They're as boring as hell, and they are insensitive, really very difficult to justify in terms of their long-term sensitivity. So what we need is agnostic real-world data, and this is where I think the wearables come in. This is where artificial intelligence, I get very excited about artificial intelligence in that context, where you can take data. Let's say we inject a particular promising therapy into the knees of our patients, and then we don't ask them to complete questionnaires. We just look at our activity, okay? We don't rely on their perception of pain. We simply rely on what movement they undertake and the steps they take and physical activity or quality of sleep as indirect or even gait. Analysis of gait and movement could be really sensitive endpoints. None of these have been proved, but that's in the direction in which we're heading. I think those are going to be very promising, particularly for those drug companies trying to develop and market orthobiologics, stem cell therapies, gene therapies. Those are the most promising biomarkers rather than the same old questionnaires. But the same old questionnaires are tools that we are already working with. They're in our hands. But we have to recognize their weaknesses and recognize that maybe there's something in the future that could be much more sensitive and agnostic in terms of looking at positive outcomes, or like an algorithm. It may not be necessarily one or two particular biomarkers. It could be a signature combination of those. And I think that's what AI has the potential to deliver.
Nice. Nice. Yeah. I was actually speaking with a medical doctor last week, and he actually brought up secretomes, and he was telling me, he's like, I mean, I've heard of them before, like in when I took orgo chem or some type of biology class in college, but I haven't heard of that in a while. And so when he brought it up, I was like, I looked it up briefly, and then you mentioned it. And why is there such I guess an increase in secretomes now?
Um, it goes back to cost and complexity.
In that
So
interest.
it's going to be cheaper than a live cell therapy platform. or a gene therapy platform. These are very expensive, very sophisticated technologies. Secretome is, if you have a cell culture facility, you can culture the cells either in adherent format or in suspension, and those cells produce proteins. They produce growth factors. They secrete things and put it out into the medium. You can take that medium, you can concentrate that medium, and that becomes your secretome, your therapeutic secretome. And it has a number of vesicles in it, extracellular vesicles, exosomes, and they're also part of that secretome. So the secretome could have a number of different components, but the membrane-bound extracellular vesicles, the exosomes, will be not only bearing on the surface a number of proteins, but also inside them, there will be growth factors and cytokines and microRNAs and things that could reprogram cells. But of course, we need to recognize that you could have an exosome and that's, I want to go back to the birth tissues versus the senescent tissues. So potentially your birth tissues could have a secretome that's very different in terms of its biological properties to the secretome of a much older donor. And we need to recognize that. So cost and complexity are simple answers to your question. But there's a
Interesting.
lot of excitement now. A lot of funding is, I don't know the situation in the US, but in Europe, European Commission is funding a lot of projects in this area, and there's a lot of interest and activity. My own team, there's some collaborative connections we have with colleagues in Austria and colleagues in Israel and Germany who are very interested in exosomes and secretome. So watch this space, Oscar. It's very promising, and I think could be a very disruptive innovation in orthobiologics. It's part of the orthobiologic arsenal. It's always been part of the arsenal anyway.
Yes, I agree. I agree. I actually, put what's a secretome, and it's a complete collection of signaling molecules like growth factors, cytokines, peptides, and extracellular vesicles released by cells. So essentially, they got pretty much almost everything that PRP has. They have exosomes in there. They got a couple of peptides. So
And
it pretty much
safe
complete
too,
package.
because it doesn't have any capacity to change the DNA in your cells. It doesn't have the machinery to do anything disruptive like that. But it's very, very different from a live cell therapy, because with every cell therapy, there are risks. So look, with anything you inject into the joint, there's risks, right? What's the most obvious risk? The most obvious risk is actually infection, right?
Yes.
Anything you inject into the joint, even if it's PRP and hyaluronic acid or derivatives of hyaluronic acid, the biggest challenge is prevention of infection. And as long as you conduct the injection in a clinic, in a properly certified space with proper air filtration, and you swap down with alcohol, etc., you minimize the chances of infection. But what about infection from viruses? Now, cells could carry transposable elements. Now, this might be a very difficult concept for some non-scientists, but all of our cells have evolved for many years from our ancestors, right? A lot of the cells that we have in our bodies have potentially a number of elements that could be transposable elements, digital signatures from the past. That could be from viral infections. And the viruses are real. There are retroviruses. There are numerous different families of viruses that have infected humans, early humans, and our ancestors long before Homo sapiens were evolved. Some of the early humans, every living thing has had to coexist with bacterial pathogens and with viruses. And so some of those viral infections, have transposable elements, DNA that can jump into a chromosome, cause damage, cause cancer, cause etc. And those are some of the things that the regulators are concerned about long term about cell therapies. They, they want evidence. They want clear evidence above safety. Above efficacy. They want evidence that treatment is safe, and it isn't going to cause neoplasia. Isn't going to cause adverse events like that. So that's why there's a very high bar with such products. You have to be able to demonstrate safety. So I guess it's the same with extracellular vesicles, with secretome. Need safety because what's the most important principle in medicine? Do no harm to the
Yes.
patient. The most important thing is making sure the patient's safe, and so we need to develop therapeutics that are over and above everything else, safe, and then efficacious. And so that's what we need to do, develop the evidence base for that safety and efficacy.
Yeah.
But
No, I
so
agree. I mean,
the riskier treatments are gene-based, viral gene therapy. Very promising, very, very promising, but risky because there are lots of unknowns. And I don't know if we've got enough time in the last 15, 20 minutes of this conversation to dig deep into that topic. But gene therapy involving adenoviral vectors, adeno-associated viral vectors, or some of the other viruses that can potentially cause disruption to the cell are much riskier. That's why there's a much higher bar in terms of their regulatory approval and acceptance by the FDA and the European Medicines Agency. And so, but they're still very promising because we've got the technology now. We have learn how to manipulate the viral vectors. We can take genes and insert them into these. We have CRISPR, enzymes, we've got mini CRISPR enzymes. We can do very sophisticated genetic engineering, but ultimately the gene therapy, again, has to be over and above everything else, safe and then efficacious too. So there's gene therapy, which we haven't had much time to talk about. And I think there's a lot of interest in gene therapy. And there are currently, we published a paper on gene therapy platforms in, I think it was September or October 2025. We're following that up with a more comprehensive review of all the cell therapy platforms. But right now, the most popular gene therapy platform is adeno-associated virus because it's thought to be much less conspicuous to the immune system, especially for injection into the knee. And there are several ongoing phase 2 clinical trials looking at the efficacy of adeno-associated viral vector gene therapy for osteoarthritis, targeting either interleukin-1 receptor antagonist, the natural signaling antagonist for the IL-1 signaling pathway, or some chondrogenic genes that can activate chondrocytes, key chondrocytes, if you like, rejuvenation and chondrogenic factors. But most of the exciting trials right now are focused on countering the catabolic inflammatory activity. So it's interleukin-1 receptor antagonist. Very promising, but of course, there are critics. There are critics of these platforms too. Again, the cost. I had a conversation with my team, and I joked with them. It's a little bit like, there's a fly in your office, and it's really irritating you, but you don't have a fly swatter to kill it. You can't reach out and find the newspaper to kill this fly. Instead, you reach for a cruise missile and you terminate this fly with a cruise missile. So gene therapy, some very, very strong critics of gene therapy say, well, it's a little bit like killing a fly with a cruise missile. It's a very expensive therapy. It's over the top. And do we need such interventions? I think it's an interesting debate. Cost, we know with all these platforms, constantly comes down. So everything starts really expensive. With all the biological drugs in rheumatology, all the antibodies in the early days, 20, 30 years ago, they were super expensive. And then gradually cost comes down, production platforms become widely adopted. And I think cell gene therapy, biologics, orthobiologics, costs are high, but future is very bright because costs will come down as our capabilities for developing and um, ensuring consistency in these biological factors improves. So that's my very glass half full optimistic appraisal of what will happen in the future. But of course, there are many hurdles in terms of regulatory hurdles and demonstrating true efficacy.
We'll definitely have to do a follow up podcast with first with gene therapy and get deeper into
Absolutely. Yeah. The gene therapy topic needs probably much more in depth, much more critical, deep diving. And I think for rare diseases, very rare lethal diseases, is a completely different scenario because we now have several examples where rare diseases in children have been treated effectively and safely with gene therapy. But it's a very rare disease, and there was a very low hanging fruit in terms of testing it in a patient that was literally going to die, and the therapy saved them. That's very different from demonstrating efficacy of a biological in the knees of an elderly population. Totally different ballgame.
interesting. Um, so you were talking about regulatory, and I didn't ask that question when we were talking about how the Florida stem cell bill, right, makes it a little bit easier for physicians here in Florida. Um, what about for physicians in other states that don't have, a state bill or something like this to protect them, right? But they have, the patients are asking for it. They have, high demand, like which, what we were talking about earlier. Um, there's a lot of physicians and providers using these birth tissue products, right? Either as Wharton's jelly, exosomes, and the FDA currently has been, very straightforward with how they see these products. And one of the most common concerns questions that I see is that I'm going to lose my license, right? I'm going to, I can't do that, right? So what would be some, I guess advice or some good pointers for a physician that wants to innovate? They see these doctors, the patients that are asking for these patients, like, how to navigate that, right? Because I understand if I were a physician, I wouldn't want to lose my license that I spent, 20, 30, 20 years building it, and obviously continuing that. So yeah, what would be a good, what would be a good advice for them?
Work and operate within the uh, best legal framework in the state in which you live and work. It's not the Wild West, it's not Butch Cassidy and the Sundance Kid, and it's not um, the classical Wild West with cowboys shooting in every direction. Um, Moving towards FDA approval is complex in orthobiologics, right? We know the
Yes.
challenges, but this shift from this concept of the Wild West to state-regulated clinical frameworks, like Florida's new law, it's like a stepping stone. I see it as a stepping stone towards, it's a bridge to possible full FDA approval. Now, look, the US is going through unprecedented social political change right now. I
Yes.
know the administration, and I'm going to say this as diplomatically and as neutrally as possible. The current administration is really shaking things up, right? That shaking up is happening across all government departments, and I suspect the NIH and the FDA will be further shaken up in the coming months and years. Who knows if we will recognize them in the future? Look, I'm British. We had a disastrous thing in Britain called Brexit, and we're still trying to adapt to some of the uh, terrible things that happened as a consequence. But putting Brexit aside and just focusing on the reality of the regulatory pathways, they're to protect patients, they're to protect consumers. And I think the best way for the doctors to operate is recognize the language the FDA expects. Do not use, be fully transparent about using an off-label treatment. Be fully transparent about uh, not providing a cure. Some companies may be using that kind of terminology that this is a cure for this disease or that disease. That's where the challenging, potentially legally complex cases may arise, because in orthobiologics it's much simpler. It's a much more different scenario, and full transparency is really important. Look,
I
I'm
agree.
involved in one study. Uh, in the wrap up minutes, I just say I'm involved in a study. It's a stem cell trial of a product from Israel, and it comes from uh, a company that established the placental stem cell therapy, and it's actually several products for several different conditions. And so you have to recognize it's a big world out there, and there are different regulatory agencies, and uh, the FDA's position on birth products is clear, but there are other countries and there are other territories where there's active, very buzzing research and development on birth tissues. And it's actually a stem cell platform, particularly for osteoarthritis, that we are testing. We're not in a position to provide it. It's not approved yet, and it won't be approved for a while. But the research and development is crucially important, must be done. Why? Because there is an unmet medical need, because osteoarthritis is a serious disease. All the osteoarticular conditions are serious diseases. We don't have solutions and drugs, and we need to innovate. And some of those innovations require mistakes, and we will learn from those mistakes. And some of those innovations, I have no doubt, will be very disruptive. And time will tell which of those disruptive innovations um, will succeed. If you ask me, Ali, so what do you think the next treatment for osteoarthritis will actually come from? I suspect it actually won't be from the orthopedic research community. It's going to be a disruptive treatment from another discipline of medicine. Remember that that's how the very first biologics for rheumatology came from. They were not from rheumatology research. It was actually from cancer research. All the TNF inhibitors, they actually came from basic cancer research. So, but what I'm really saying is we need to be collaborative and multidisciplinary in the way we approach uh, orthobiologic treatment and osteoarticular diseases. There are 200 plus different forms of musculoskeletal conditions. We can't treat them all with anti-inflammatory drugs, and we need more precision tools. And some of those precision tools could turn out to be gene therapies, and some of those precision tools may turn out to be uh, extracellular vesicles, exosomes, and secretome. Time will tell, but we have to be open to possibilities.
We do. And then you mentioned actually a good point there. So I know you're you're worldwide, right? And so different countries have different regulations and things like that. From your knowledge, what country do you think has the most spelled out um, regulations when it comes to birth tissues? Because when you look at the FDA's regulations, it's just the 351 or the 361, right? But then with all these products and everything, the Secretomes, Exosomes, everything, it's just different. they're each classified differently, right? You have cell factors, you have a lot of different things. So like, from your knowledge, which country has the, a good classification system, I would say, from your
I'm
knowledge?
going to answer your question in a slightly different way, not directly answering it to you, but look, you've got the FDA in the United States, you've got the European Medicines Agency in Europe, but then you've got the specific countries that have very specialized regulatory structures. So in Germany, you have the Paul Ehrlich Institute. You've got the Japanese Food and Drug Administration. You've got the Korean FDA. You've also got the equivalent bodies in China and Hong Kong. I love Asia, and all my friends and colleagues know how much I adore Asia, not just because I have some Asian genes ancestrally mixed into my gene pool, but because it's always been for me an area of the world to go and learn about. Curiosity to learn about a very different part of the world. But when it comes to cell and gene therapy, South Korea is a model country. They have the strongest platforms for cell and gene therapy. They actually have a very, very tough FDA, very critical FDA, but they've also got the most extensive network of tissue banking, biobanking. And so it's a very, very different environment in South Korea, in Taiwan, in Japan, in Singapore, in Hong Kong, compared to the United States, compared to uh, Nordic countries, Scandinavian countries, the UK, Germany, France. Every country is very different. But Asia is where there's genuinely a different feel and a different kind of vibe in orthobiologics. Cell gene therapies are very advanced there. And I think you need to recognize these big geographical differences.
Interesting. Why do you
And
think there's a difference in that?
some of them are doing their trials in places like Australia. So they go to Australia and they actually conduct their trials in Australian clinical trial centers. And it's again because they take advantage of the local rules. They fully understand the rules and the regulations and the frameworks in which they have to operate. And there might be different bars. Some of the bars may not be so high. And I think this is where we should look to the East to see what emerges from Asia in terms of therapies in this particular market, in this sector.
Yeah, I agree. I mean, even though, they say that the United States can be, advanced and all that, but I think even us, we have a lot to learn from other countries and vice versa. I feel like every country can learn from any country, right? Doesn't matter what country you are, right?
Yeah,
How
absolutely.
advanced. Yeah. Yeah.
Ultimately, is we have to serve. I'm a public servant. I don't work for a private institution. I work for a publicly funded institution. And if I receive funding from um, government of Finland or European Union, my funding is coming from the taxpayer. That
Mhm.
makes me a public servant. It makes me directly responsible to the patients. I need to make sure that the research that I conduct is as relevant to them as possible. So I make sure in everything that I do, I consult the patient organizations. I work with several of them, particularly the one in the Netherlands, Ruma Netherlands. I consult for them. I get involved in their think tanks and work with the patients that so get to know the consumer, get to know the patient, understand their needs, fully
Yeah.
understand the regulatory framework. It's not perfect anywhere,
No.
and
Nope.
recognize um, recognize opportunities for where things could be tried and tested. So I know I haven't answered your question. All right. I deliberately haven't answered your question. I haven't told you which country may have a very particular low bar for birth products, but I've given you a flavor of
Yeah.
variations across the
Yeah.
different parts of the
Yeah.
world.
No, I appreciate that. I guess a low bar wasn't really what I was asking, more for like a classification that, that actually fits each product, right, in a way, right? Because I think, having a low bar may, the way I'm looking at it is, it may not be good, because then depending, if it's low bar, then any drug or product or anything can just go through, right? And then is it safe? Is it effective? So I'm all for high quality, high setting high expectations and high bars, right? It's just more of like a good classification, right? Like setting things because I'm sure, like what you were saying, like how gene therapy might is riskier, right? So having obviously a classification for gene therapy that okay, you have to go through, I don't know, 10 years, whatever that bar is, versus secretomes, you said, right? It's like it's cost effective. They're not that risky, right? So having a different classification for that type of product, right? So that's what I kind of meant.
I would say look to the Korean, South Korean environment, because they're really setting themselves up really well for these kinds of innovative therapies. And I think it's because they've put so much money into setting up the infrastructure needed for um, stem cell therapeutic development and implementation. Not just develop it, but once it's developed, actually implement it and deliver it to the patients in large numbers. So you need to have the infrastructure for that. So not just the research, but the the ability to deliver it. I think Asia is more prepared, and
Interesting.
especially post-COVID, there's even more preparedness in the Far East compared to Europe and the United States.
That makes sense. So I guess the last question and final word is where do you see I would say stem cells in the next 5-10 years? Where do you see that?
So a number of ongoing trials need to provide readouts for us in the coming few years, but let's not forget that the most successful therapies need to be effective, safe, and cheap, right? Ultimately, somebody has to pay for it. So if a treatment comes in from a secretome platform, which is much cheaper, not so complex to store and deliver, doesn't require that level of high level complexity, of course, there's going to be wider adoption for a cheaper, more robust platform. So maybe it could be a stem cell derivative rather than stem cells themselves. But we might have some publications, high level publications coming in from the scientific community in the coming months and years to say, well, no, actually, it has to be a live cell because there is a particular nuance that a live cell would provide that a secretome and extracellular vesicle secretome platform may not. So the answer to that is not yet known, but I suspect it's again horses for courses. If you want to navigate a particular type of course, you have to select your horse and your cart very carefully. You don't, select your Arabian racehorse to go up mountains and steep valleys. So it's again, classical expression, horses for courses. Select your tools and your platforms very carefully. But who knows? It's possible that maybe cell therapy for some orthopedic conditions and maybe secretome or extracellular vesicle therapies for others, as long as, above all, it's safe and efficacious, and then cheap, affordable, and disruptive.
Yeah, that's well, thank you so much for that. That's something that I've never really heard of, right? The cheap specifically, right? Safe, effective, yes, but cost effective, but you are
Economics is so
absolutely right.
important. Absolutely, economics is so important.
It
You know,
is.
you don't want to develop a treatment that's going to cost um, $60,000 because you have to compare it to one gold standard we haven't talked about, which is knee arthroplasty. And you got to calculate the cost, the payer's cost of a new knee. And that's one gold standard you can compare to. Treatments have to be far cheaper than that, really, because we know that some people will have to get arthroplasty. There's nothing else that's going to help them. uh,
Yes.
Either full or partial arthroplasty of the knee is, end game for some people who have very advanced arthritis. But again, some of the exosome, cell-based, secretome, exosome-based treatments could be coming in much earlier in the disease journey, in the patient journey, in the disease pathogenesis and progression journey, and potentially save the joint from arthroplasty. And then that's where the big saving comes from. If it's been well costed, if it's a treatment that's not costing thousands and thousands of dollars, and it could potentially delay arthroplasty by 5, 7, 10 years, that would be an amazing innovation.
Absolutely right about that. Um, yeah. Okay. Do you have any final words for the audience? What's any inspirational?
Please contact Oscar with some feedback, and if there's any
Yes.
topics that we haven't discussed and you'd like Oscar to delve into in more detail with another guest, or myself, or even have a debate, please let Oscar know by contacting him on LinkedIn. And thank you, Oscar, for doing everything that you do. You are uh, certainly stimulating a lot of open debate and conversation on this platform.
Thank you so much for that. I've been trying, and it's very much needed, right? Good debate, respectful debates, talking to each other. Um, Thank you for saying that, and I really appreciate
My
it.
pleasure. The next conversation we have will be private, and there will be a couple of tequilas, margaritas on the table.
For sure. All righty. Well, I hope you guys have a beautiful day, and I'll see you in the next one.
Happy
Adios.
New Year, everyone. Thanks.
Happy New Year, everyone.